Hello, anatomy students. Thanks for sticking with me. This is the third and final part of the lecture on the male reproductive system. And here we're going to talk about sperm production. Now, there's going to be some mention about cellular division, both mitosis and meiosis. And I created a brief video that gives you an overview of those two processes. So make sure that you check back in Canvas. If you haven't watched that, you might want to pause here and just go back and see that just so that you understand what I'm even talking about. So anyway, here we go. This is, we're going to talk about sperm production. We are going to talk about spermatogenesis, and then also just a little bit about spermiogenesis. And so the two words sound the same, but they do mean different things. So spermatogenesis starts with spermatogonia, which are cells that are activated during puberty, which is when a male gets a very large burst of testosterone to the system. The spermatogonia are going to undergo mitosis, which is cellular division that results in two daughter cells that are exact genetic copies of each other. One of those daughter cells will remain a stem cell inside of the seminiferous tubule, and it will continue to divide in order to produce primary spermatocytes. Notice that both of these daughter cells are 2N. They have the diploid number of chromosomes, which is 46. Then the primary spermatocyte is going to undergo meiosis 1. And remember that before this first meiotic division, there are a couple of things that happen to ensure that the final daughter cells are going to be genetically unique from each other. So they undergo meiosis 1. We have now 2 haploid daughter cells. So remember that that first meiotic division is reductional in nature. So you're reducing the number of chromosomes from 46 to 23. These are called secondary spermatocytes. And then it is the secondary spermatocytes that undergo meiosis 2. That produces 4 daughter cells of equal size, and each of these daughter cells is genetically unique. These are called spermatids, and these are the cells that are going to undergo spermiogenesis in order to produce mature sperm. So the spermatogonia are starting in the outside portion of the wall of the seminiferous tubules in the outer layers. This is the outermost portion, and then as you move towards the lumen is the space that's found inside of the tubules themselves. So as the cells move from the outer portion of the wall towards the lumen, they are maturing, they are growing. And so as development progresses, they move towards the inside of the lumen, so that then once, and these, remember, are not fully mature sperm, but they sure look like them. Their maturation process isn't complete until they get to the tail of the epididymis and the vas deferens. But here you can see that we're progressing from a spermatogonium, and then after that first mitotic division, you have the primary spermatocyte, and it's moving in towards the lumen of the seminiferous tubule. And then this primary spermatocyte undergoes meiosis 1 division to produce the secondary spermatocyte, and then the secondary spermatocyte undergoes meiosis 2, and we get the spermatids. And then it's the spermatids that are going to undergo spermiogenesis in order to form the spermatozoa, which will then enter the seminiferous tubule and then migrate down to the tail of the epididymis. It is nurse cells that are found in the tissue of the seminiferous tubule that are going to help the spermatocytes along in their journey. So nurse cells are pretty large cells, and their cytoplasm surrounds the developing sperm. And so here you can see that I've got these arrows here. They're pointing to the cytoplasm of these nurse cells. Here's the nucleus of the nurse cell. And so the nurse cell is going to use their cytoplasm to support the development of the developing sperm. The nurse cells are going to affect mitosis, meiosis, and also spermiogenesis. So FSH and LH are going to stimulate the nerve cells into action. So remember that these two hormones are coming from the anterior pituitary gland. They are the gonadotropins. FSH stands for follicle stimulating hormone and LH stands for luteinizing hormone. And these are released from the anterior pituitary, and they are going to stimulate nerve cells in the seminiferous tubules to support mitosis and meiosis. Nerve cells also support spermiogenesis, which is the development of the spermatids into cells that look like mature sperm. It's the development of the head, the neck, and the tail of the sperm. But the nerve cells are also controlling how the speed at which cellular division is taking place, and that's going to do that with negative feedback. So the nerve cells are going to monitor the amount of new sperm cells that are being created. And when the nerve cell senses that it needs to slow down that synthesis, then it's going to secrete inhibin. And inhibin will feed back to the hypothalamus. So we'll get to that on the next slide. But the other thing that the nerve cell does is it secretes an androgen binding protein, an ABP, and that is going to attract and bind to testosterone, increasing the amount of testosterone that is present in the seminiferous tubules in order to further the maturation of the new spermatids. It's going to support spermiogenesis. All right, so as I said, the nerve cells are going to secrete inhibin. So let's take a look at that process right now. So we're going to just start at the beginning. So we're going to talk about the hypothalamus, which is releasing GnRH, which is gonadotropin releasing hormone. Gonadotropin releasing hormone, and guys, this should be review from our endocrine unit. Gonadotropin releasing hormone from the hypothalamus is going to stimulate the release of LH, luteinizing hormone, and FSH, follicle stimulating hormone, from the anterior pituitary. That gets into the bloodstream and stimulates the nerve cell to support cellular division, to increase cellular division of the spermatogonia, of the primary spermatocytes, of the secondary spermatocytes, all of those different divisions that are required in order to create the new spermatids. But then as cellular division increases, the nerve cell might be like, oh, we've got enough of this. We can kind of slow this process down. So the nerve cells secrete inhibin, which is going to not only inhibit the anterior pituitary gland, but it's also going to inhibit the hypothalamus. So inhibin is going to slow down the release of LH and FSH, but it's also going to slow down the release of GnRH from the hypothalamus. So inhibin is going to have kind of a two-pronged function here. As a result, we have decreased division of spermatogonia and spermatocytes. So as I mentioned, spermiogenesis is the development of sperm, which are actually highly specialized cells, even though their structure looks a little bit simple. These are tiny. They have very few organelles. That's going to make the sperm very light, able to move very easily. And it also allows the sperm to carry out its function, which is to deliver genetic material to an oocyte during fertilization. And what we're going to learn later on is that an oocyte becomes an ovum once it has become fertilized by one of these cells, by a sperm. The sperm lack glycogen, which means that it needs the fructose, which is found inside of the semen, which is released from the seminal vesicles. It needs the fructose from those seminal vesicles for energy in order for it to move. So the head of the sperm is basically a packet of genetic material. It's the nucleus. It contains DNA. It's also got a packet that contains an enzyme. This packet is called the acrosome, and that's essential for fertilization. So in order for a sperm to break through the lining or the envelope of the oocyte, it's got to break down that tissue. And so that's what the acrosome is for. It carries enzymes in order to do that, so that the genetic material from the sperm can combine then with the genetic material that's found inside of the oocyte, and allow fertilization to occur. The middle portion, the middle piece of the sperm, which contains the mitochondria, is connected to the head by the neck. The neck contains centrioles, and this is where microtubules that form the flagellum is going to begin. The middle piece of our sperm is the engine of the cell. It contains mitochondria, which is going to produce ATP from the fructose that is surrounding the sperm in the semen. It's going to produce this ATP so that the flagellum can propel this sperm through the urethra during ejaculation into the female reproductive tract and then up to the fallopian tube in order to perform fertilization. And then the tail, as I said, is made of microtubules. It's called a flagellum, and its movement is whip-like or like a corkscrew. Sorry, you can't see it, like a corkscrew kind of movement in order to propel it through that reproductive tract and up into the fallopian tube. All right, these are my references for this presentation. Thank you very much for watching. And again, you can fill out that question sheet just to make sure that you're understanding the very main points of this 3-part presentation. And I'll see you guys in class. Bye.